Okay, last set of slides for cellular respiration ever. This is, so it says the title is wrong. So the title of this slide is wrong. It is about what happens when you don't have oxygen present, right? Because we talked about how this is mostly an aerobic process, needs oxygen. What happens when you don't have oxygen? Because that happens. And so we're going to talk about that. I've only been in my classroom talking to myself for like an hour and a half. But these are all done. This is for the week. And then probably next week you will have a test on photosynthesis and respiration. So this is the summary of aerobic respiration. It happens when oxygen is present. We make a yield of about 30 to 32 ATP. What happens when we don't have oxygen? Without oxygen, the electron transport chain will cease to operate. And so the first two steps of cellular respiration can occur, but chemiosmosis and therefore the production of basically 28 of my ATP are not made. In this case, glycolysis couples with two different things, either fermentation or anaerobic respiration, to make ATP. Anaerobic respiration uses an electron transport chain where the final electron receptor is something other than oxygen. And so the excited electron that normally would split the hydrogens off, it is fueled by something other than oxygen. The other thing that I compare with is fermentation, which uses substrate level phosphorylation instead of an electron transport chain. So it gets rid of the need for an electron transport chain, period. Therefore, it phosphorylates ATP or phosphorylates ADP into ATP in a totally different way. There's two types of fermentation. Fermentation consists of glycolysis plus reactions that regenerate NAD, which are reused into glycolysis, either alcohol fermentation or lactic acid fermentation. Alcoholic fermentation, pyruvate is converted into ethanol in two steps, and it releases CO2 first. This is pretty much only used by yeast to and then put into stuff to make beer and wine and bread and things like that. But it's the way that yeast make their own energy is alcoholic fermentation. If we look, basically on the left side is alcoholic fermentation. Basically what happens is after glycolysis makes pyruvate, instead of making acetyl-CoA, it makes ethanol. In the lactic acid fermentation, pyruvate is made into lactate instead of acetyl-CoA. This is going to be alcoholic fermentation up close. I still make 2 ATP, and I create my NADH with my hydrogens attached. I just make ethanol instead. If we look at it in this way, basically the glucose is split down into pyruvate, and we see that instead of going into acetyl-CoA, it makes ethanol. This is lactic acid fermentation. The pyruvate is turned into lactate instead. In lactic acid fermentation, pyruvate is reduced to NADH, forming lactate as its end product and not releasing any CO2. Lactic acid fermentation is used by fungi and bacteria, and it can be used to make yogurt and cheese. Human muscle cells use this to generate ATP when O2 is scarce. This is why the longer you work out and you start going into oxygen deprivation, you're just not getting enough oxygen, even if you're breathing really hard. It's why you'll build up lactic acid and end up getting sore. Lactate fermentation is basically this process. We've already looked at this image. And these aren't super important to know, just examples of what happens without oxygen. Fermentation compared to anaerobic and aerobic respiration. They all use glycolysis too, and so they're still, or glycolysis, and so they're still producing 2 ATP. They, in all examples, so these are the similarities. They all use glycolysis and they make 2 ATP. In all three, NADH is the oxidized agent that accepts electrons or becomes NADH. The processes have different final electron acceptors, which means that they enter into the other two parts of cellular respiration differently. But cellular respiration produces 32 ATP per glucose. Fermentation only produces 2 ATP because it has nothing to send to the Krebs cycle and to the last part. And so it just keeps going around and around making 2 ATP. It's very ineffective, obviously, because I could be making 32. But this is why oxygen is a very important part of cellular respiration, although it can be done without it. So there's certain organisms that only do anaerobic respiration. We call them obligate anaerobes. They carry out fermentation, and they can't survive in the presence of oxygen. They can only do it this way. Yeast and other bacteria are what we call should be facilitative, but facilitative anaerobes basically means that they can survive either way, but they will adapt to do fermentation when they need to, when they're not in the presence of oxygen. In this, pyruvate is basically the fork in the road. If they can make it, it'll continue to make 32 ATP. If it can't, it'll stick in its fermentation cycle and just make 2. And that should say facilitative, FACI. Right, so we see here, if I end up not having oxygen, if I'm like a yeast cell, I will make ethanol and lactate. I'll go through my fermentation process. I'll make 2 ATP, and I'll continue that cycle. If oxygen is present, I will make acetyl-CoA, which in turn is used to fuel the rest of the cycle, making 32 ATP. The significance of this we've talked about, but ancient prokaryotes are thought to have only used glycolysis even before there was oxygen, because glycolysis happens whether or not oxygen is present. And so it is kind of the first energy producing process that we see. Questions that come up, but I eat more than sugar, right? We always talk about glucose. These pathways funnel electrons from all different kinds of organic molecules into cellular respiration. And glycolysis accepts a wide range of carbohydrates. So we use glucose as an example, but I can use carbs. Proteins are digested before being used, but they all end up being broken down enough to be used for these processes. If we look at it this way, carbohydrates, so sugars, can be used directly into glycolysis. Proteins have to be broken down to be used. Some fats, like glycerol, can go right into glycolysis. Fatty acids have to be broken down a little bit first, but they all end up getting incorporated at some point. The regulation of respiration. So I need oxygen, right, to get the maximum amount of energy. I use a homeostatic feedback system, which basically tells me that if I'm starting to drop below a certain amount of ATP, my respiration speeds up. When there's plenty of respiration, my breathing will slow down. It's why you start breathing heavily when you are working out. It's when your breath really slows down when you're sleeping. The control of this is based mainly on regulating enzymes at strategic points. So I don't actually look at the level of ATP specifically. I probably look at the level of how quickly ATP synthase is working. So that is it for cellular respiration. You will need to study photosynthesis and cellular respiration for Monday.